Alright, next we're going to talk about protein specifically because they're a little more complicated as far as their structure and function is. Like I said, the functions of proteins, the list is a mile long. We're going to talk about the bond here, peptide bond, and that is between amino acids. Okay, peptide bond is between amino acids. The most important part of these amino acids, like I said, is the R group, which makes the amino acid what it is. And the reason that the R group is also important is because it gives the protein its final shape. And the final shape of a protein is very complicated. If you take take like a really long noodle that you boiled in water. Okay, you boiled a really long noodle and you just simply dropped it on the floor. It would take this really weird squiggled up shape. It would dry and it would have that shape. That's kind of like what a protein does. It just really folds on top of itself, really complex folding, takes the shape, and that shape determines what that protein does. Here's that amino acid that I showed you on the first side of amino acids, by the way. This is tryptophan, and some people are going, oh yeah, tryptophan, I've heard of that for something about Thanksgiving. Quick side note, not on the test, but guys, there's no way the tryptophan in the turkey is causing you to get drowsy. This is the thing that people say, oh yeah, tryptophan, that's what's in the turkey that makes you drowsy. No, it's because you just ate more than you've eaten in the last 3 days combined, and now your body's trying to catch up processing all this food. It takes energy to process all that food, and your body's getting worn out just by sitting there because you just ate so much. You just stuffed yourself so much with all this food. So it's not the tryptophan. You'd have to eat like an elephant-sized turkey to get close to the tryptophan effect. Okay, it's just the amount of turkey that and mashed potatoes and everything else that you ate. But anyways, here's the peptide bond formation, which you can see in those animations on the previous slide for protein. But notice there's an OH right here and an H, two H's, but one of these H's is going to combine with this OH and drop out as a water, and then that connection is going to be made. That peptide bond is going to be made. Same thing over here. Enzymes, a special kind of protein. Enzymes work the same way proteins do. Their shape has everything to do with their function. Okay, enzymes are proteins. They're just a special kind of protein that acts as a catalyst. You can see speeds up metabolic reactions, things that are happening within our bodies. And so just take a look at this picture. Know what's going on here, because you will see something just like this again and again, probably a couple times. Here's the enzyme down here. Notice it has a very special shape, kind of like a puzzle. Okay, whatever it is that fits into it, because this could be a starch molecule or some other carbohydrate. Okay, that's going to be acted upon by this enzyme. It's going to break it down into its products. And this enzyme can go off and now find another starch molecule. Okay, and then go break it down again. Okay, we're breaking down starch to get energy out of it, or a plant would be. Okay, so the enzymes in the plant, this enzyme here, breaking down the starch. And actually, amylase in your saliva is the enzyme that breaks down starch. And we'll see that a little bit later this year. Here's a graph, or sorry, here's a table, because you're going to have to look at and interpret data tables and graphs for enzymes. Here's a table showing you 6 different enzymes, A through F. These don't really mean anything. This is just made up stuff just to kind of make my point. Here's a pH range that each enzyme works at. And then here is a certain temperature that each enzyme works at. Again, this is all just made up stuff. These enzymes don't exist, but maybe these ranges are close to some enzyme. I don't know. But in any case, you can kind of look through these, and there's a question for you. Which enzyme would work best in the human body, and why? Okay, I'll give you a hint. It's not this one down here, because check out the temperature. Okay, 400 degrees is pretty hot. That would disintegrate most enzymes we have in our bodies. So your correct answer, you're looking for a neutral pH and a temperature that is within the range of our bodies, and that should fall under letter D right here. Neutral pH, somewhere around 7, and our body temperature is about 98.5. Here's the range in which this enzyme would work in our bodies, the human body. And then towards the end of our presentation here, this is simply just a graph made from this table on the left, and just information you should be able to pick out of here. Okay, you can see this enzyme starts to work a little bit better, the activity, meaning that it's working. It's working a little bit more efficiently, or more of the enzymes are working at this pH here, somewhere around 7.5 or 8. Okay, and pretty neutral. And then a rapid decline, just like the incline on the other side, once it gets past that point. Okay, so the enzyme activity is best at around 7.5 or 8. The optimal pH for this enzyme, whatever it is, enzyme A or B, whatever from that table, could be any of them, that is within this range of pH, anywhere from 6.5 to 8.5 is within the working range for this specific enzyme. And then again, same graph, but to make the point that there is a range for all enzymes, both for pH, temperature, and you can see that this enzyme stops working at a certain point, 0 somewhere over here. When an enzyme falls apart, it's called denatured. That's an important vocab word to remember. It falls apart. Okay, if you heat something up too high, think of something that you heat up too much, you can make it fall apart. An enzyme is that thing. Okay, if you heat it up too much or increase or decrease the pH too much, the enzyme is simply going to fall apart. Let's go back to our noodle that we dried up on the floor. If you were to put that thing back in boiling water, it's going to undo the shape that it had. It's going to go back to a noodle looking shape, kind of, just a squiggly mess. It's not going to be this like hardened formed structure of like a loop or two loops. It's going to go back to being just a kind of strung out noodle. Okay, that's denatured, heating it up. Good example of denatured proteins are, or enzymes, are in eggs. When you put an egg on the frying pan, raw egg on the frying pan, it starts to turn white. That is evidence of the proteins in there being denatured. They're actually changing color, and their function is going to change too. They're not going to work correctly. Okay, so denatured is when an enzyme or any protein falls apart. The pH is out of range, or the temperature is too hot. That's what causes an enzyme to be denatured. So that's the end of the show. Guys, the most important parts are water, carbohydrates, nucleic acids, proteins, lipids. There's 5 parts of the test. What they look like, what they're made of, and how they function. Okay? Properties of water, how water functions. Carbohydrates, what they're made of, how they function, and very importantly, nucleic acid structure and their function. Genetic information is going to be very important later on this year, and proteins too. Structure and function directly related. And then again, the lipids, triglycerides, and phospholipids.